Hi, everyone. Thank you for joining our third Synthetic Biology and Control Theory breakout session. So this one is a little bit shifted from our previous ones. This one is more focused on application of synthetic biology and health applications. And of course, the topic of pandemics. This is something that I'm kind of interested in. How can synthetic biology assist in sensing, preparation, and recovery for a pandemic? So we have two hosts. Professor Katie Galloway is from MIT School of Engineering, where she is the W.M. Keck Career Development Professor in Biomedical Engineering. Her interests span systems biology, synthetic biology, genetic control systems, and oncology and metastasis. Also hosting is Dr. Brigitte Sanders from NIH's National Institute of Allergy and Infectious Diseases, where she's a program director. She's also interested in pandemic preparation and, of course, how synthetic biology can fill some of the gaps that we have in all aspects of pandemic preparation. So before I pass the virtual mic to our host, we'll do a virtual poll on the system. So Heather can pass this to you.
Jamon, can you also introduce Dr. Reed Chapman?
Oh, sure. Reed is also from the NIAIDS as well, where he is a program director. And Reed, I'm sorry, if you could provide your focus areas.
Oh, you got it correct. Focus areas are systems biology and certain aspects of virology.
Okay, great. Okay, Heather.
Great. And in just a moment, we're going to have a word cloud on the screen for you. You can either use this QR code or Holly is going to place a link in the chat box that you can use.
So it looks like we have a good spread of research areas. So we can wait another minute to see if anyone else pops anything else. So yeah, this is kind of what I expected. So this kind of helps set the stage for us. So with that, I can pass it to, oh, we're getting some more. With that, I can pass it to Katie and Brigitte and Reed.
Wonderful. Thanks so much, Dermot. Yeah, to get going, I'm just going to share some slides that we prepared to kind of structure our time. While I'm doing that, maybe let me see here. It's always simpler when you're not having to do this live, right? Here we go. I'm hoping you're seeing my slides. No one's saying no, so I assume the answer is yes. All right, so today we're delighted to be able to continue the breakout sessions for the Synthetic Biology Consortium Meeting, and hopefully what we really want to do today is get your guys' feedback and bring you into the discussion of how do we respond to a pandemic, and then what are the opportunities specifically for synthetic biology in preparing, in responding and recovering. So today, I'm going to tell you a little bit about our time, and then I'm going to especially turn it over to Brigitte to talk about the pandemic response that she's an expert in. But we want to give a little bit of background motivation, discuss some of these different, the topic areas we'll try to cover today. We'll see where the discussion leads us. We hope it'll be somewhere really exciting. We'll have lots to then report back on. That's our goal, is to have some summaries for the meeting the following week. Basically this will be that we will have, instead of a breakout, because we have a smaller group, we're able to have then a discussion all together. There'll be four different discussion topics. And then we will be wrapping up by 4:45 so that you guys can be on your way. And that's sort of going to be depending on how the conversation goes. It might be shorter, it might be long, slightly longer in places, but our goal is to be done by 4:45. So synthetic biology, I think one of the exciting things about synthetic biology is, the opportunity to program biomolecules and cells. That is to then think about the cells, using cells as factories. And this is a lot of, it was birthed out of metabolic engineering, but synthetic biology has done a lot to contribute to this, especially with respect to thinking about dynamic control, and that you can't just engineer these cells alone. You have to think about how to optimally engineer them. Minimal systems, of course, building different biomolecules and figure out what is the minimal system in which they can function. And then I think very excitingly thinking about cells as these autonomous systems. And really, how can all of these things contribute to our ability to more rapidly and effectively respond to a pandemic? To really lead that discussion, we have Dr. Brigitte Sanders, who's going to tell us a little bit about the NIAID Pandemic Preparedness Plan, and also Reid Shabham, who I believe will be taking us through the following slides on the NIAID Pandemic Preparedness Plan. And Reid, let me know if you need me to move to the next slide.
Sure, perfect. Sounds good. So I'll talk about the next two slides. Thanks for the opportunity to just quickly go over these. And I'm just the messenger here, not the curator, but I think it's important to take a quick note about the following information. So we're all aware of what happened with coronavirus over the past couple of years, and there were certain successes and there were certain limitations. And so NIAID has developed a pandemic preparedness plan with the specific goals in mind to sort of enable our response in the future. And those include to systematically characterize pathogens of concern and increase research and surveillance to identify threats before they emerge. So proactive action around specific pathogens, shorten timelines between pathogen emergence or outbreak onset, and the authorization and approval of candidate diagnostics, vaccines and therapeutics, which is right in the line with the NIAID mission. And then to bridge or eliminate existing gaps in research, infrastructure, and technology, and expand preclinical and clinical testing capacity. And I imagine we'll probably get into more details about this in our discussions. So the next slide. This is a nice little graphical illustration of kind of what NIAID's been thinking about over the last couple of years. So this was released about a year ago in December of 2021. And what you can see on the far hand left side are, I should backpedal a moment and just say that the pandemic preparedness plan really centers around viruses. The division got together or the institute got together and sort of thought about what are potential pathogens of concern that could cause a pandemic. And those were whittled down into a variety of different viruses, different viral families. And I may forget all of them, but for example, you have the coronaviridae, you have the Bunyiaviridae, you have the Arenoviridae, you have all these RNA viruses mostly that have the potential to spread and emerge very quickly. And the idea here was to select prototype pathogens based on predetermined criteria. And so the idea is that within each one of these viral families, there would be a specific representative pathogen or a prototype pathogen. And we might know something about that individual pathogen and how it might take that information and apply it to other families. And so for one example might be a specific Lassa virus would be a specific virus that we want to understand a little bit more about in the Arenoviridae family. And understand its binding and entry, what antigens are important for vaccines, et cetera, et cetera. And you can imagine how synthetic biology would feed into some of those studies. Then the next phase is sort of basic preclinical and translational research. So that's in the middle of the slide, where you're really taking a deeper dive into pathogen biology, immunology, pathogenesis, et cetera, developing critical reagents and animal models, and really sort of fully understanding what's required to develop vaccines, diagnostics, and therapeutics against that prototype pathogen. And then the ultimate goal is to move those promising medical countermeasures to clinical trials, on the right-hand side here. And the idea would be that you might have phase 1, phase 2 ready vaccines, monoclonals, drugs, synthetic, that could then be leveraged and applied for a pandemic outbreak. And the idea being that if an arenavirus emerges and it's not Lassa, but it's a cousin of Lassa, we would understand the fundamental principles of what's important for a vaccine, the fundamental principles of what's important for a small molecule to inhibit the replication of that pathogen. And one could then going back to the previous slide, you could then shorten timelines for response. And so I think that's sort of a summary of those two slides. Go to the next slide and I'll hand it back over.
Great, thank you so much. And even just so that kind of leads into our next slide is where we've been very familiar, I think, over the last few years with the idea that that mRNA vaccines fill a critical need in our pandemic response. And I think we all have really experienced the opportunity and the privilege of having that response, relatively quickly. And whether this is going to be sort of what we continue with, I think is part of the question that we're here to talk about today, in terms of how do we innovate, and then also integrating into thinking about innovating from a synthetic biology perspective. Even just sort of this diagram here is meant to illustrate that there's various ways in which we can alter vaccines themselves. And of course, this isn't the only thing that we would consider in terms of a pandemic response. Again, these parts and pieces really feed into this idea of what synthetic biology has been very good at, which is abstraction. And this idea that there are tools, parts, and systems that basically are composed together to get different behaviors. Again, it can be a molecule or it could be a larger system. And it really goes back to that fundamental thing that's inspired a lot of synthetic biology, this idea that we're oftentimes trying to create things so that we know that we understand them, whether that's a vaccine or a cell type. And of course, we've come many years from the 90s when everyone was just fusing GFP to various things and asking, can we measure something? And is that measurement useful in some sort of quantitative way, and which it could be used for various forms of screening or measurement. And really, I think that that's where the engineering and the biology meet, is where we can ask, how do we measure things? How do we do this on a single cell level? This has inspired, I think, a lot of people to think about expanding into single cell measurements and using cells as sensors. That's something that my lab is very excited about, this idea that we spend a lot of time thinking about building gene circuits, and the ones that are very interesting to us, at least, and I think increasingly to everyone who works in the mammalian synthetic biology space, is using circuits as these modules that could then plug into the cells that are sensors to be able to understand disease processes and report information out. So tying GFP to something that's more interesting in terms of drug screening. And that we think this is a very powerful modality because it allows us to build systems that are then going to span a range. So not just 2D culture, where a lot of our disease modeling has existed so far, but potentially being able to then take circuits that can sense disease and disease processes across multiple scales, from organ on a chip, organoids, and all the way up to human on a chip. And I think this is especially important as we think about pandemics, when we don't actually know oftentimes which tissue is going to be the most vulnerable to the different types of viruses or what's going to emerge. And this gives us then the opportunity to look at across multiple different cell types. With coronavirus as an example, we know that damage to cardiac and neural tissue is especially important, and the targeting of those tissues is very important. And then, of course, understanding the disease functions, whether it be in the brain or whether it be in the heart. Again, potentially we can model these and capture relevant disease processes by building gene circuits to track that, and in theory do this drug screening that I alluded to in a more rapid way. In the meantime, of course, we're oftentimes dependent upon mouse models, which sometimes can confound identifying therapeutics that work for humans. On the other end of this, just besides drug screening, we'd like to be able to actually repair these tissues right after damage. So something I think increasingly we'll see from the synthetic biology community is going in to engineer cells that would actually have a therapeutic function, reprogramming tissues to replace damaged neural tissue, to regenerate, and potentially even surveil and look for disease. And so this is something that our lab thinks about a lot, again, in terms of using this for disease modeling. Could we understand how pandemic is going to affect neural cells? Can we screen for therapeutics? And then long-term, if there is neural damage, especially, or cardiac, in these non-regenerative tissues, can we actually deliver therapeutics? So these are some of the ideas we wanted to put out there as we discuss. And for our time, these are just ideas, but really we want to think about how can synthetic biology help us prepare and recover from a pandemic. We're going to spend the first bit of time discussing the preparation aspect, which is so important, and real-time acute response. Then we'll go towards, what can synthetic biology do for towards drug screening, whether in terms of mitigating disease or potentially preventing it or repairing. And then finally towards, when we are subject to a pandemic that has induced damage, how do we actually repair? The goal here is we're going to have a discussion. We'll have a scribe who's going to summarize some points. I'm going to take those slides and those points and summarize them into slides that we present at the workshop next week. And this is our general schedule, which also you should have a link to in the chat. If we could put that up one more time, that might be useful. Because I think we have a few participants who joined since I added that link. So we're going to have a little bit of an intro. We have a small enough group here that we can do and have a nice discussion. We'll have discussion on topics 1 and 2. I'll introduce those in just a second for about 15 minutes each, but we'll see how the discussion basically moves and whether the discussion should expand or contract. We'll take some time then to summarize for maybe 10 minutes, just to kind of, say, okay, we've had this discussion. What do we want to come away with summary points? Then we'll have a break at just 5 minutes before the hour for 5 minutes. We'll come back and switch to topics 3 and 4. And then again, we'll have a wrap up and we'll try to have you guys on your way by 4:45, if not sooner, depending on how the discussion goes. For our topics areas, these are again in that document, which hopefully you're now linked to in the chat, you should have access to. So topic area 1, again, preparing for a pandemic. This is obviously the best way to have a fast response to a pandemic. So I think spending time here is well worth our time. So how can we do that now that especially we realize the need in a way that I think, more people are aware? What do we need? Biosensors, diagnostics. How do we remain nimble? I think we've had a really nice introduction by Reed on terms of some of the things we should think through. We'd like to think more on that. Topic 2, real-time acute pandemic responses. So in the midst of a pandemic, which we have or have not prepared for, how do we respond? Topic 3, are there things we can do for drug screening? This is, I think, also a real-time potentially response, depending on how well we do with our drug screening infrastructure. And then how do we, on topic 4, think about, again, responding long-term to restoring health after people have already experienced a pandemic. So that's basically what our time's going to look like today. And we're really looking forward to getting your perspectives. And to start with, again, we're going to go and we just would like to know who you are. What's your research interest? And then we'll start the discussion on topics 1 and 2. For now, I'm just going to stop the share and hopefully we can see your guys' faces. I didn't introduce myself too much, but just to start with, I'm Katie Galloway. I'm interested in synthetic biology, and I'm really interested in using gene circuits to do disease modeling and drug screening in the context of the specific cell types that are affected. So using cells as sensors, and that's why I'm here. Nice to meet you all.
My name is Jermat Chen. So what I'll do is I'll just point at someone or I'll mention their name and then they can give their brief background. So I'm a program officer and I work on the synthetic biology portfolio at the Institute of Biomolecular Imaging and Bioengineering. So this fits well within our wheelhouse. So Reed, why don't you go next?
Sure. I'm Reed Shabman. I'm a program officer in the Division of Microbiology and Infectious Diseases. There's 3 different extramural divisions at NIAID. And so I fall into that category. Yeah, and I'm a molecular virologist by background. Currently, I have a portfolio for systems biology where we sort of do multi-omic integration and generate predictive models from that.
Okay, Brigitte.
My name is Bridget Sanders, and I'm a program officer in the 2nd division, the Division of AIDS, and my interests are in HIV and TB. And I also have a strong interest in synthetic biology and how synthetic biology can help with infectious diseases in general, especially with pandemic preparedness, identifying the agent and, providing a response as soon as possible.
Okay, apologies if I get your name wrong. Atur?
That is correct. So I'm a research scientist at Northeastern University at the Center for Theoretical Biological Physics. So I work on gene regulation. I try to get the gene regulatory network and how the model the gene regulatory network and how that drives the system.
Okay, great. Taesuk?
Do you work on any infectious diseases or mostly on gene regulation?
Currently, I'm working on immune response in zebrafish.
Okay. Do you work with low Bolux by any chance?
No.
No,
I-
but he's
I
at
was-
Northwestern too, is that right?
Northeastern.
Are you at Northeastern?
Yeah, in Boston.
Oh, okay, I see. I'm sorry.
Okay, Taesuk.
Hey, how are you? So I'm Taesuk Moon. I'm the WashU professor, but today I'm representing EBRC. This is Engineering Biology Research Consortium that is the nonprofit organization to provide vision to the global community and US government, especially synthetic biology or engineering biology topics. And my expertise is basically mostly bacteria and including probiotic. But I've been in synthetic biology almost 2 decades, but I actually entered mammalian synthetic biology last year. But I still my main focus is probiotic engineering and biosensor and biocontainment.
So that was Washington University, St. Louis?
Yes.
Okay.
Yang.
Hi everyone. My name is Xiaoyin Wang. I'm a program officer at the NCI's Cancer Imaging Program, and we're new in terms of trying to incorporate SynthBio into our program. We're particularly interested in intersecting molecular imaging and the SynthBio tools and approaches for cancer diagnosis and therapy. Thank you very much. Looking forward to learning lots of new things.
Great.
Welcome.
Brian, can you give us your background?
Sure. So I'm Brian Key. So I work at Columbia University. I work in the Virginia Cornish lab. So I'm actually a grad student, but I'm in charge of a big division in my lab. So so a brief introduction of what Cornish lab does. So we translate engineered yeast like Saccharomyces cerevisiae into biosensor therapeutics. And for me, I started the division of vaccinology. So I'm making vaccines for SARS-CoV-2 and Candida-related infections and other infectious diseases. So I'm here to learn more about NIH-related fundings, especially for my project. So
What type of vaccines are you producing?
So we're engineering like probiotic yeast as the vaccine. So it's a oral vaccine, and the antigens. So we're trying to develop SARS-CoV-2 and Candida albicans and other pathogens as well.
As prototype vaccines, and then you can go on from there. Okay.
Yes, exactly. Yeah.
Okay, great. I think that's it. Katie, I can turn it back over to you.
All right. Yeah. So I think we just want to start off with our topic 1, which is going to be the preparing for a pandemic. And I think that's so everything. I think that Reed really actually should be probably also facilitating this discussion, because I think between Bridget also as well, whatever you would like to sort of say in terms of adding to this topic area that we've already sort of outlined a little bit, like biosensors, diagnostics, identifying threats. Is this something, how are we going to approach this problem? Is this going to be real-time monitoring of wastewater? We're doing this in Boston all the time. Do we need to start expanding what we're looking for? How do we prepare, how do we prepare and try and just in terms of surveillance, but then also what do we need to do in terms of stockpiling, developing infrastructure? What did we not do? And what can we learn? These are some of the questions that just came to my mind. Again, I'm quite unfamiliar with actually the response other than what I've been watching on TV and reading. So I think there are good things and good tools that we've seen come online fast. I'm guessing part of that is we had infrastructure in place, but what are we missing? Maybe we could start the conversation a little bit there, but feel free to refine that in terms of what we've seen already and like what we, would be able to know and do better next time.
Well, I think we have come a long way. You know, if you think about the AIDS pandemic, which the first cases really were reported in June of 1981. And then it took really 3 years to identify the agent. I mean, can you imagine having a pandemic going on for 3 years before you know even what the agent is? I mean, nowadays that would be completely unacceptable. I think we already made tremendous progress, but I think it really does start with the clinicians. I mean, for example, with Zika, there was a very smart Brazilian doctor that noticed an unusual amount of microcephalies in her clinic. You know, I mean, she said she sees microcephalies from time to time, but there were just an increase in cases. And she reported that, and that alerted to the problem. Because if you monitor wastewater or, you know, the environment, I mean, it's non-specific. There's so many viruses and bacteria everywhere. The question is, what's coming through? But also with SARS-CoV-2, an unusual amount of respiratory infections were reported, but I think it was suppressed initially. But in Brazil, I don't think it was suppressed. I mean, the clinician reported that, and then they followed up on it, and they discovered that Zika was the cause. So things actually, you know, moved very quickly with both Zika and SARS-CoV-2. Would you agree, Reed?
Yeah, I would agree. I was looking at our Google sheet to see, to tee it up. I completely agree. I mean, Bridget makes a great point about the gap in HIV along, you know, back a while ago to identify the etiological agent. I mean, I guess you can, thinking about synthetic biology and bringing it back to those areas. I mean, well, I guess take it aside from the diagnostic standpoint. So we'll take that. I mean, the fact that we are able to generate millions of genomic sequences at light speed now is been accelerated by the pandemic. I don't think there's any disputing that. And so how does that help? Well, that allows us to look in molecular detail at the sequence of the virus, and then allows us to determine what's circulating and make the appropriate matching vaccine or therapeutic. You know, I think the Zika example is another interesting one because it's, you know, post-HIV, pre SARS-CoV-2, and I think that you know, that was an unusual one because that virus was originally identified in the 40s, the 1940s, and then kind of in Africa and Malaysia, I believe, and then, you know, a newer version shows up in South America. So, you know, you're not, I guess that illustrates the fact that you're not always expecting to find certain things. The Ebola outbreak in 2013 on the west coast of Africa, you know, Ebola was always in the DRC and Sudan and Uganda. It wasn't on the west coast. And so folks were looking for Lassa fever, for example, right? And so having agnostic diagnostics to find things is very important. And that's, you know, so I think continuing to invest in technology development that allows you to pick up things agnostically through, you know, different mechanisms is critical. And I guess I could go on to therapeutics and vaccines, but maybe I'll pause there and see if anybody else has any thoughts on these topics.
May I go?
Please.
Yes, go ahead.
So, as I said, I entered mammalian synthetic biology last year, partially due to one of my former students who joined Pfizer and developed COVID vaccine at Pfizer. And of course, I'm very proud of him, but I didn't expect my student working on vaccine because my lab is still, you know, at the time was the bacterial synthetic biology lab. And then I realized I could contribute to the society by doing something. But to me, the important thing for us to develop is basically, I mean, this is my kind of my question. I mean, for example, you know, can we detect any infectious disease, including virus or bacterium transmitted or fungus causes ones, using a generalizable sensor platform? I mean, if we can answer that question or develop something, there might be, something we could say we are prepared for whatever pandemic in the future. And then another thing I could say is, another question is, can we develop a device which can be placed in an office, house, theater, hospitals, football stadium, whatever, and detect all the current and future viruses or multi-drug resistant pathogen or dangerous chemical biological weapons? If that is possible, that will be another, huge thing. We are not there yet, but I guess today's, discussion probably about, discussing or coming up with those kind of, technological possibility, I guess.
I still think there's a huge gap though for bacteria and parasites, and I can speak from my own experience. I was at the doctor on Friday and they couldn't find really what I had. They just could say I didn't have COVID and I didn't have flu, but it's probably some bacterial infection and they just prescribe some antibiotic, but it's pure guessing.
Mhm.
I mean, they don't really have very good diagnostics for most common diseases.
Mhm.
It's mostly flu, RSV, COVID, but it's true, it's more for viruses. For bacteria, they never do diagnostics, except for TB, which also most doctors' offices I don't think have. And then they prescribe some antibiotic, and that's why we have so much antibiotic resistance, I think,
Mhm.
because the antibiotics are prescribed just on best guess and not on scientific data.
That brings up so the pandemic that I remember before this one was the pertussis outbreak. We had a huge pertussis outbreak in Southern California when I was living there. And pertussis is obviously not particularly dangerous to adults, but mostly, but to children is quite bad. And of course, the lapse in vaccination or extensive vaccination was one of the causes, because there's especially in Southern California, there had been pockets where people had particularly not been getting vaccinated. But I was surprised to see how long it actually took for them to diagnose whether they should give you antibiotics for pertussis or not if you came in with something that was like whooping cough. Where this is something that I think we still have this enormous lag, as you're saying, Brigitte. Like our rapid diagnostics at a point of care are pretty limited. And so given what you were just speaking about earlier, in terms of, doctors being really the front lines of being able to see pandemics evolving, that seems like a big blind spot.
It is, totally.
And so until you actually have then really gross manifestations of a disease, there's nothing to do. And it seems like the other challenge is identification isn't enough alone, if only because it sounds like many of these viruses were already existent for a long time before they became really public health concerns, in the sense that this is shutting down entire regions and things like that. So what's the gap there, I wonder, in terms of being able to say, not only do we have this virus circulating, but it's now acutely problematic? How do we even how do we know? You guys probably know the answer to that. I don't know the answer to that.
I mean, that's a great point. To me, I mean, that's why I'm actually sort of proposing ambitious or crazy idea. Can we make a device that can be placed in just your office, I mean, your houses, or theater, whatever place, rather than visiting doctor's office, doing whatever lengthy process to, diagnose, you have the infected by whatever the bacteria or virus. Can we make those kind of device? I mean, of course, that's too ambitious right now, but with the technology development, like, paper-based one, and actually, I'm also developing something based on the organoid. But that's mostly focused on the virus and RNA sequence or DNA sequence, not the bacteria. But once we demonstrate something, we could expand the same idea to the other bacterial species and the pathogen as well. But of course, that takes time.
Sure.
But that should be done.
No, this is, I'm gonna, so Tessa, imagining, I guess this is what I want to ask Reed and Brigitte, or anybody else on the call who's I'd have an answer for this, is that level of surveillance enough?
It's a great question. So there are a couple of things here. I mean, I think it's important to note what is available. So companies like BioFire have rapid diagnostic panels for known bacteria and viruses.
Yeah, they're very good. I actually have experience with BioFire. I can only recommend them.
Yeah, and yeah, so exactly, yeah. So there are some out there. I think one challenge with infectious diseases is, and I'll try to put it back on some technology, is that if you don't actively have a viremia or bacteremia or something like that, it's hard to catch it, right? And so that's always a challenge. And so Brigitte's example of, not being able to find it, there could be a number of different reasons. So one other area could be, looking at host response to determine if you have something bacterial or something viral. There's technology development in that area as well. And I think, there's, to Katie, to your point about what's out there, BioWatch was launched a long time ago, I believe, looking for, should we be sampling wastewater? Should we be sampling the environment? Should we go out and swab every bat that we can find? What's the right approach? And it's not an easy answer. I think, through technology of being able to do next generation sequencing and informatics to look at sequences and then match them to our databases is a really great technology. And if we can implement that to saying, into better diagnostics to find pathogens, I think that's an area to explore. I think one big challenge there is, and I don't know, is regulatory, right? So can you, there's sort of two buckets, I guess. There's, can we do it, and then can we make it so that it can be, of clinical use.
So here's my vision. So here's my vision. What I thought. It generally doesn't make sense to go to the doctor before you have the diagnostics. And, sometimes the diagnostics takes too long. So you get sick, you have your kid at home, you swab yourself, you drop it off in a box, in 24 hours, you get your diagnosis, and then you go to the doctor and get examined and get your treatment. I mean, you don't even go to the doctor until the results come back. Unless you are like super sick, but in the most ideal situation, as soon as you have the first symptoms, you take your swab or whatever, kit you have for different, body fluids. You drop it off outside in a diagnostic lab. You don't even contaminate anyone. And then you get all these super diagnostics, like look for 100 different things because they have the kit that TSOC developed and, the machine, and they can look for all these things. And, then you get your results by email or some confidential fax or, some modern technology. And then you know already what you have, most likely. And then you go to the doctor prepared, with all your results. That's sort of my vision, which would, I think, save a lot of money, a lot of time, and also would, make the treatment more targeted.
Yeah, I like it. So, but to be clear, my device will not be available until a few years later. But my vision is actually, of course, I mean, I wanted to kind of make people just kind of look at whatever color of device or whatever device I'm developing, and then saying this room is contaminated, and then you just need to evacuate or do something cleaning. That's my goal, because in that way you don't need to worry about even being infected or potentially infected, immediately when the room is contaminated by whatever virus or disease. And that's what I want to do. In that way, we could, also minimize spread of the virus infection to neighbors or some other people, but that takes time because we should kind of develop some device that detecting, not only COVID, but some other virus as well. But that will require lots of research effort and funding, of course, but I need to propose something right now because we are collaborating with other people, collecting some preliminary data right now, but of course that takes time.
So I think that so two modes we've kind of seen, I think here is we have, so diagnosing people as they get sick, and then also spaces, thinking about how do we look at spaces as
space.
Whether it's airborne,
Yeah.
I mean, disease, can you have monitoring there that makes sense? And then also, I guess, the wastewater. So you're talking about, I guess, 3 primary sampling routes, which are a little bit different: people specific, air spaces, and then probably water spaces. Are there any other spaces that we would look to? I don't know. Air, water, if we go to earth, like fire, but
Middle Earth.
Middle Earth.
Yeah.
Well, the microbiome is that something? I mean, I have seen people reporting on, they're like, oh, the wildlife has, X amount of COVID burden, or, so many deer in New England have, some antibodies for COVID.
Yeah.
Is that a useful metric for monitoring, or is this just sort of people's, curiosity?
Well, I would give my 2 cents to that, is that it's extremely useful because I think 70% of all infectious diseases are zoonotic in nature, right? So they come from an animal reservoir.
Do we then have to worry about spread within wildlife or even domestic animals as being like something that is going to be a second, I mean, a potential next source? Normally people think about this in terms of like maybe, food supply, large stocks of animals and stuff. Do we have to think about that with respect to wildlife or pets? Or is that very, very rare just because of the numbers?
Well, I think tigers are a problem for SARS-CoV-2. Like, I think that Roy got infected by his tiger, but not many people have tigers. So
I'm
I
sorry,
think,
who got infected by a tiger?
do you know Siegfried and Roy?
Really? Yes,
Yeah,
no,
I think
know.
that I think he got infected by his tigers because they were positive.
Wow.
But that's just what I think, but it's hard to prove. But I mean, yeah, I think for SARS-CoV-2, I think humans are the biggest transmitters. I don't think that pets are a huge risk, but on a one-to-one basis, you can get it from especially felines, I think.
I've seen some dogs walking
Cats.
around with masks, and I was wondering, I couldn't tell if it was sort of meant to be adorable or they were actually thought that there was a protective layer needed and or beneficial. Are animals affected in the same way? I mean, probably not, but I didn't know. I did see, I mean, this was in Cambridge. I saw a guy walking with a poor dog with really big mask, almost like a diaper.
I think that tigers can get sick, and cats, but not to the same extent as humans. I have to look at some literature, but in general, I mean, it depends on the virus or bacteria, like for HIV, it's not transmittable to animals, so it's not a source for HIV, but for Ebola, yeah, like bats, can be sources. We know for coronaviruses, bats are sources for coronaviruses. With influenza, I mean, animals are huge. Huge source. You remember the swine flu? I mean, the first Spanish flu came from pigs. In Kansas, they call it the Spanish flu, but it actually originated in Kansas. And so pig farms. It depends on the virus. If the virus is very promiscuous in their glycans and tropism, then animals can play a huge role. But some viruses have very narrow host ranges, but you almost immediately have to be careful. When there's a new virus, you have to be careful with animal transmission. And then, of course, we have the whole range of mosquito-borne viruses, the so-called arboviruses, which are indirectly transmitted to animals because the mosquitoes bite the animals, but it just depends.
Interesting. This
Yeah.
has been a super, so do you have anything, sorry, Reed, you were going to say?
I just, one real quick point on that is, I think sort of taking it back to the synthetic biology space is there's clearly a lot of places that you can get infected. You can get infected from other people, you can get infected from animal sources, and you need to sample all those areas. One place with synthetic biology, so others on the call that know more about this than me, but maybe you could imagine as a diagnostic, you could make synthetic material that would be representative of these infectious diseases, viruses, bacteria, fungi, whatever it is, and you could put them into your diagnostic. You could customize that quickly, depending on your approach. And so you could, when it might be more higher burden for humans, but if you're going to look in the animal livestock area, you could potentially apply those more readily, without the regulatory burden. So I think I'm curious of others, I guess there's like CRISPR-based diagnostics and all these other types of diagnostics, but if it's a nucleic acid-based technology, you would imagine that you're going to need gene cassettes, a wide range of them to put into those, so you could quickly customize. So I think that's where synthetic biology is helping, and it's sort of moving us to that place a lot faster.
Very good.
I would say though that the response with SARS-CoV-2 was pretty optimal. I mean, in less than a month, they knew the sequence and how fast it developed the vaccines and the diagnostics. I still do think that the human factor is the biggest problem. People not realizing, fast enough how dangerous a situation can be. I'm not sure whether you can improve that with synthetic biology. I think people knew that there was SARS-CoV-2 around, but they didn't take it seriously enough, or maybe it didn't make it down into, every community. I mean, if you think what happened in New York, I mean, realizing if there's an outbreak in Washington state that the virus is probably already in New York. I think that kind of messaging was, I think, the biggest delay. I'm not sure how synthetic biology can help with that, because I'm not sure how much more optimal you can have the response. I mean, there was so much money poured into SARS-CoV-2, and the speed with vaccines were developed, and I think the biggest hurdle is the FDA. That's another huge step, to get the FDA apprised of the synthetic biology efforts. So it doesn't take a huge time to approve synthetic biology products, because, if you develop them very fast, but then it takes the FDA 2 years to approve them, it's not very helpful.
Yeah. Brigitte, you are just perfectly on time for thinking about our topic too. We did say we were going to take a break at 3:55, but the conversation is so nice right now. Unless people feel that they really need a break, I suggest that maybe we continue our discussion on topic 2. As everyone, I can only see a few faces, half your guys' faces. So unless, I think we will continue on that. And because again, you're exactly like this real-time response, how fast we could actually make things and get approval by the FDA is a huge part. So there's a regulatory component, there's a science component. And I think that the thing we're all, have seen didn't work out entirely perfectly is also then the human response to uptake and, recognition of the danger of COVID, and then also the opportunity to then take as much protection through the vaccine. I don't think we can do anything necessarily too much about the human response. I think we're still working that out with the sociologists, but maybe we could turn our topic to topic 2 and talk about how do we develop these synthetic vaccines to be fast? Is mRNA technology something now that we can rapidly deploy on new strains and variants of concern? It seems like the answer to that may be yes, but are there challenges where we don't expect mRNA to be the best, or are there things we need to think about, maybe cold storage deployment to low resource areas? Maybe that's where we can kind of turn some of this discussion towards that real-time acute pandemic response for topic 2. And I'm not an expert in this either, so I'm asking questions.
Reed, you want to take that, or?
Sure, but I can start, but I'd like to also call on anyone else that, I think there were some folks on the call, I'm sorry, that were talking about vaccine development as well. And so feel free to come on and comment. I mean, I think, mRNA vaccines are a great success because we're able to go from sequence to synthesis pretty quickly, lyophilize if you need to, ship it somewhere, reconstitute and go. So I think it's a huge success. I think the question is, how fast can we make it? And so, you know, to use like an influenza example is, by the time you identify the subtype of flu, you know, by the time you actually make, it takes about a year to make the current flu vaccine. And so if we could implement a synthetic vaccine for influenza, you might be able to, you know, accelerate responses, get better matches. There's a lot of potential there. So I think it's a huge place to be. I think one thing to be determined is the longevity and effectiveness of RNA vaccines, mRNA vaccines. You know, I think we all experience that now. We're to get a boost every 6 months. How durable is that response? You know, is it due to the nature of the virus, or is it due to the technology? But not that that's sort of a side note. But I'm curious what others think about this in the space. Is there anything beyond mRNA vaccines and synthetic biology that you could use, or if anybody has any other thoughts?
Well, I want to comment a little bit on here.
Yeah.
So since I've been working on this, and I attend a lot of conferences, especially I met like the creator, Katalin Carrico and Drew Weissman. So they really point out 2 points. So, you know, 20 years ago when they first started developing mRNA technology, one of the greatest challenges is that they cannot get funding. And, you know, so I remember they presented like almost for 10 years and they haven't get any R1 grant. And I think, you know, for NIH, perhaps like the early stage development of platform vaccine technologies like really needs to be addressed, especially for like synthetic biology. One of the greatest advantages that we can create like new platforms that can potentially address. So the second issue they brought up is the affordability and the scalability of the vaccines. So, you know, there are limitations for mRNA vaccines to be like how affordable, like can it be as cheap as like $2 per dose or less than that? And, you know, for example, when I'm like the platform I am developing right now is addressing this issue, and we can kind of predicted that to be at least less than $5 per dose or less than that range.
But your vaccines are oral, you're saying?
The vaccines are all?
No, they're oral.
Yeah, they're oral. Yes, that's
okay.
correct.
Okay. I mean, I personally think the mRNA vaccines are less than ideal. The biggest advantage is that you can produce them so fast, and that they're cell-free. But you know, there's problems with storage. There's problems with acceptability. There's the problem with the formulation, the lipid nanoparticles, which cause huge reactions in some people. I personally think on the vaccine part, there's a huge room for improvement. You know, to have not only vaccines, but to have a platform. I mean, what I think we should strive towards is to have a platform where you just switch the inserts, you know. You already have a prototype vaccine that's approved by the FDA. And then if a new pathogen comes, you just switch the insert and you don't have to go through all the approval process again, just similar like with the new bivalent SARS-CoV-2 vaccine that wasn't tested in clinical trials. You already have the platform and you just, you know, put in a different sequence. You know, that would sort of be, I think, the most ideal situation.
So people have brought up the mRNA technology. I guess one of the questions I've been thinking about is, you know, as Brian and others mentioned, that's, you know, fairly, I don't want to say old technology, but it was developed back in the 70s-ish when people were doing a lot of work on it. My question is, what tools do we have now that are like what mRNA was back then that we can use for these things? You know, this is looking into the crystal ball to see, you know, who's the next superstar, or But are there things out there that people are working on that are interesting? Because sometimes they'll just pop up, like CRISPR popped up because, you know, CRISPR wasn't designed to do cuts. It was designed for something else. And they said, hey, this is interesting. We can apply it for X, Y, and Z.
I mean, I'm not expert in RNA vaccine development, but I heard a lot from my student who joined Pfizer and developed COVID vaccine. But to me, now we have the technology, really the platform technology. In that way, we could change the sequence of the whatever RNA to develop another vaccine, probably very quickly. But the key problem, especially in terms of public acceptance, that is basically reduce the side effect. And then what I heard from him is basically side effect coming from the other component, the component other than RNA, something like, whatever, other one, those things we probably need to improve. But I guess, that is more likely, I don't know, not necessarily synthetic biology contribution, but somehow try and error or multiple combinatorial testing somehow with the, I don't know, some useful animal model. That might be something we should address, but I believe now we have the technology, platform technology, in terms of RNA part, but the other component we should be changed. You should change them, otherwise we may have another side effect, because that is kind of one barriers, not all the people can accepting the vaccine.
Yeah, I think a huge problem is also that people only seem to be working at high speed if there's a pandemic, But I think we really have to work when there's no pandemic, mostly, and then, be prepared. But the way the funding always works is that, everything's just accelerated if there's a pandemic, and then everything slows down, which is like really the opposite, like it's sort of like the stock market. You're supposed to invest when the stock market is down and sell if the stock market is up. But intuitively, most people do it the other way around. And it also seems to the same cycle, with the funding. I mean, people don't work very hard on a problem if it's not there, but as soon as the problem comes back, when it's actually too late, then people start working again. But I think what we need to do now is develop those prototype vaccines and then, have platforms ready, and the same for therapeutics.
Sure. I mean, that's why also I'm very welcoming or very optimistic when Alpha H start, because I guess, their role, the Alpha H role should be something kind of nurturing something very ambitious, and then something like we can kind of have the breakthrough technology developed through that funding mechanism.
Yeah, I'm very excited that Julia is still on. I see that David left, but Jamont, you might know too. We had a meeting, a SynBio meeting last year, and one of the companies that was featured at the SynBio meeting was Ginkgo Bio.
Yep.
And now the head or one of the persons from Ginkgo Bio got chosen as the ARPA H director. So that's really very promising.
Yeah, I know her very well. Actually,
Oh, you do?
Pardon?
You know her well?
Yeah, I know her very well since she was a DARPA director.
He was a DARPA program manager for.
Yeah, she was a DARPA program manager and then joined the Ginkgo Bio.
I see.
And then Ginkgo Bio worked for the founder, actually my friend, because we went to MIT together and we basically met every week to discuss the synthetic biology future, and then they decide to choose, the startup area. I decided to choose faculty member, and in fact, I might be their first employee because they approached me saying, "Tessa, do you want to join my company?" And I said no, unfortunately. Unfortunately,
Well.
yeah.
If I could, can I make just to get on the record for Jermont, just 2 quick points. So one is on technology, a gap in technology with synthetic biology. And please, everybody correct me if I don't have this right, but just printing DNA, right? So like chemical synthesis, we can make 200 MERS, hard stop. And with enzymatic synthesis, you can make like 60 MERS, right? And so like if you want to make a synthetic SARS-CoV-2 spike, which is 3,000, and you go to GenScript and you say, give me that right now, they'll say 2 weeks. And so where there is a technological gap for printing long DNA, it seems. And I know that DARPA is working on that. And you can stitch it together, obviously, as we do, but wouldn't it be cool if we could build it longer, faster, de novo? And then the other area real quick is synthetic antibody development. So, could you understand what antibody is important, and can you print that antibody sequence and express it properly to make a therapeutic? So I think those are a challenge and an exciting area.
But we haven't heard much from Jia Yang. Do you want to say anything, Jia Yang? Is that how you pronounce your name? I'm sorry if I mispronounce it.
No, you pronounce it perfectly. I'm sorry. Oops. Sorry, I wasn't. I mean, I don't think I have the much of a background and know how to provide something that is useful. But like I said, I'm here to learn. So all this information, I'm trying to absorb it and see how it applies in my area of research, but at the same time trying to think of things that what we've been dealing with might be fitting to the discussion. So not that I wasn't listening, I was actually I have been paying close attention. I just don't think I have anything useful to say at this moment.
I just wanted to make sure we hear your voice and you get a chance to speak as well.
I think Yang, you might have more things to say as we turn towards also like topic area 3, where we start thinking about drug screening, molecular imaging, which may be more closer to your, and we will welcome your contributions at any stage.
Okay.
So it sounds like, printing long pieces of DNA to make synthesis, more rapid and assembling things, I think is a big deal. We can now sequence things really long. So I think always the tie-in for synthetic biology has been the longer the more easily you can sequence something, the more easily you can synthesize. I don't know if actually we still have that that tie-in, because it used to be those used to be very tightly coupled. And now with nanopore sequencing, we can actually do very, very long reads. But again, I don't know if our synthesis capabilities have scaled with that. And so Reed, that's a really good call on thinking about that level of synthesis and whether there's a gap there. And what would bridge it? I don't know.
Yeah,
I'm actually more hopeful about drug screening than about the antibodies, because I think in the beginning the antibodies were really helpful for like SARS-CoV-2, but lately the virus is just mutating too fast, and I don't think there's a single antibody right now that's working. And the effort that has been going into developing these antibodies, I think that the drugs generally target areas of the viruses that are more conserved and might be longer lasting. I don't want to completely give up hope on the antibodies, but they usually target surface proteins, otherwise they cannot work, and those are the ones that mutate the fastest. With HIV they're still using them, but they're not approved yet, and they're not using them widely. But for SARS-CoV-2 they're using them widely, and the FDA just revoked the emergency use authorization for Bebtelovimab. That was one of the more hopeful antibodies that actually NIAID developed, but the turnover is just too fast. And I'm not sure, I'm worried that the companies are a little bit You know, with the drugs, I mean, we really need drugs faster. It took a long time to develop Paxlovid, and we only really have one drug. And once there's widespread resistance, and you know, for HIV, it took a long time to develop drugs. I mean, a lot of people had to die before the drugs were there. You know, if you can create cell factories to spit out those drugs, make them synthetically somehow, I think that would be the biggest contribution. The vaccines, they were fast, but the drugs, it took a long time.
That is a beautiful, beautiful segue to topic 3. Birgit, that was just masterful. I think that you identified exactly the challenge there is like, it's the identification, right? Like what drugs are going to work, but then being able to make them in some sort of, you know, rapid fashion. You certainly don't want to be dependent upon some of the drugs which we identify, you know, oftentimes they're derived from plants, and then the extraction and manufacturing process then becomes unbelievably limited in the same way we've seen with, you know, antimalarials. And why, you know, in theory, I think, you know, for a long time, we had hoped that actually synthetic biology would be able to generate artemisinic acid or the artemisinic acid precursors, and it would be cheaper. And certainly it's a more robust way. You don't have to depend on poppy crops, but it never got quite as cheap as it needed to be. But maybe in terms of speed, we could still have an advantage, I think, if you could make drugs. But I think the challenge is identifying them and what, so this kind of turns to, I think, our next topic area, which I'll just reintroduce, although I think Brigitte giving a great intro, is just, you know, how can we use synthetic biology to improve the identification of drugs for pandemic response? And is that going to be something we're going to be using cell lines for? Do we need the exact cell types we think are affected? How would we set that up? Can we use the synthetic circuits is something that I think a lot of people are excited about, this idea about being able to triage drugs and identify candidates, make the molecules. I think one of the challenges that we've thought a lot about with in terms of drug discovery is oftentimes you have really non-specific effects, and it's difficult to then actually see, is your drug, you know, just having a general suppressive effect on cell behavior that you don't measure. But and how do we actually I think do that identification? I'm not familiar with has the process mostly been mouse-based for identifying drugs for COVID? Does anybody have?
The way usually drug companies operate is that they have huge diversity libraries. They already have those libraries available, I mean, which basically prototype compounds, and they have a lot of information on those compounds, so they already know the toxicity, the chemical behavior, scalability, and you know, if something new comes along, they use their libraries and run them against the new target.
So target-based screening, basically. So then you're still dependent upon a binding event, a very specific binding event. And so antibodies, you know, surface binding and neutralization is one way, which seems like it's you're getting around. And the second one would be then the target-based screening of small molecules against putative drug targets, proteins, RNAs. And I guess even the question of what do you target then for these viruses might be quite you know, important. And I don't know if we have a good handle on what we should be targeting or not. I'm on the sort of the other end where I'm very target agnostic, where like we use our cells as like just do phenotypic screening. And the problem with phenotypic screening is if you don't have good controls, you don't know what you're doing. And there's a lot of ways of rescuing cells and getting viability or different effects if you don't have those. And so we think circuits are really important for providing those controls, but then you need the cell type of interest normally, like an airway epithelium if you're looking for some sort of specific rescue, or a neural cell. But again, in terms of these other specific candidates, what are the how do you even identify whether you want a small molecule that would bind to a protein or to an RNA, or is it always going to be a protein?
Mostly, often with infectious disease, it's an enzyme.
An enzyme.
enzyme are the preferred, is a preferred target. If you look at HIV, reverse transcriptase, protease were the first targets, integrase. Enzyme is the preferred target because it's just the drugs work the best against enzyme. They also have drugs against certain proteins, but if you look for SARS-CoV-2, it's the RNA-dependent RNA polymerase, which is targeted by molnupiravir, and then the protease, which is targeted by Paxlovid.
So that's interesting to me because, again, is it not thought then, I mean, the sequences themselves are remarkably, I mean, the coding sequences would be potentially also targets of something like an siRNA, ASOs, or some of these other programmable molecules that we now know how to make and deliver. Is that something that people are doing? Is this something that NIAID is funding, nucleic acids as those targeted medicines? Or is there some fatal flaw in that strategy? I mean, there probably is, but.
Well, they have the so-called ASOs, the antisense
Mhm.
oligonucleotides, and there are 13 that are FDA approved. I think one was withdrawn. There's the famous story of porphyria. This is like a metabolic disease. Have you heard about it? Porphyria. It's when you have porphyrin in your blood, it's there's something wrong with your metabolic pathway, and the body produces porphyrin, which is a toxic product, and it goes in your brain and you turn crazy. And George III, the King of England, had that, and he was the king during the British American War and the Declaration of Independence. And there's a company called Alnylam in Boston, and they have a drug for this porphyrin. And they think if they would have been in place during the War of Independence, then Britain would have never lost the war because King George III would have never turned mad, and he would have won the war. But I mean, this is just a side story, but they were one of the first ones. I mean, these are so ASOs, antisense oligonucleotides. They are 14 to 22 nucleotides, and they basically target directly the nucleic acid. That's another way of targeting, but I don't think any infectious disease applications are approved. I cannot think of any, but they have metabolic diseases, children's genetic disorders. It's a newer way of targeting. I think there's also some people that are trying to target RNA with drugs as well. I think the options are unlimited, but I'm not the best person to talk about it, but you have to have target identification, then you have to do your screen, and all of this has to happen pretty fast. So I think it would be better to identify drug classes of viruses and have drugs already, prototype drugs ready, because I think drugs takes the longest to identify, not necessarily to produce, but to make a good drug. It takes just longer than a vaccine, I think.
So may I add one more point to those conversations? So I actually work for biotechnology company, 5.5 years. And then we are now talking about drug discovery or screening to prepare another pandemic. But the important limitation we have, we often forget as a faculty member. At the end of the day, really the bottleneck or rate limiting step is not necessarily discovery and others. The rate limiting step is actually approval process, including clinical trial, because that could take minimum a few years, even though you cannot go through expedited whatever process. So the technology development for the high throughput screening and so on is important, but to be prepared for the pandemic, and then the speed of the screening and so on important, but at the end of the day, anyway, that to take minimum few years. to get approved. And then in that situation, that's why I more care about diagnostic than the drugs, treatment side, because that drug will be developed, let's say 5 years later after pandemic, that is probably too late because of the approval process. So we need to kind of think about that one, and then we need to do something in a more policy side, I guess.
Yeah, that
So-
was sort of the premise with EcoHealth, where they were trying to sequence viruses in bats to see what potential pandemics could arise, and then it was sort of in pandemic preparedness.
Right.
But
That-
I
that's
don't.
it.
Hmm?
Yeah, if that is the case, I mean, I'm that's important, thing to do in advance. But how we gonna know that will be really next pandemic virus or others? That's why if that's the case, I would develop vaccine rather than treatment drug, because that might be much easier to develop vaccine than whatever drugs. So that's why, the fundamentally developing screening method with the high throughput ability will be, very important. But then practically whether that is really addressing our speed problem, that's questionable. That's my point.
So one thing maybe to follow on this is, I think that at least what we've seen from coronavirus is that, a vaccine isn't sufficient. At least one vaccine is not sufficient. And even like multiple boosters is not sufficient. So it may be for some people, the best hope is for them to survive is they get the vaccine, and it buys them some time while we're developing also small molecules or other therapeutics. And then I guess sort of what maybe is also going to be leading us here is we have about, I think, 20 more minutes or so in this discussion, and we can integrate everything together, is thinking about long-term screening for drugs potentially, or developing therapies that aren't going to just block the disease itself, but potentially repair after, or deal with cases where it's not COVID, it's long COVID.
Mhm.
And so that might be a space where, especially given the fact that it's a long and chronic condition, we might not be able to deal with the acute, but we can start screening for the long. But that's very complicated, I think, also because in those cases, do we have a target might be the challenge. And maybe Brigitte or Reed, you can answer that question, or even, I think that's a question that we ask a lot in cancer as well. Like, what's the target? Is the target moving? And then oftentimes, because you don't have a target, you don't know what that is, even with other chronic diseases like MS, oftentimes you're then still doing a phenotypic screen to try and say, well, what can we repair? What cells actually do we need to worry about? So that might be where we're going to pivot our discussion to as we kind of wrap things up, is should drug screening also then focus on repair, and is a single drug going to be enough to repair tissue, as we've seen some pretty remarkable damage. I don't know if you guys have seen some of the MRIs that people are showing from people who've had COVID, but the effect on the neural structures and stuff is pretty remarkable. And so what do we actually need to do? Is a single drug going to be enough? Will cell therapies be useful as the way for people like Parkinson's? This is thought to be the only way to potentially deal with some of those degenerative disorders. And should we treat some of these diseases that we're seeing that are coming out of infectious disease as degenerative diseases, where there might be a long connected period of inflammation that leads to degeneration? I'm curious to get your guys' thoughts on that as we talk about this last topic.
I mean, for HIV it would be really helpful. They have cured 5 patients so far through a stem cell transplant, but it's hard to find matching donors, and there's a huge risk, with the myoablation and so forth. And right now it's only indicated for people that have cancer. But if you could, have synthetic cells that could replace the immune system, I mean, you could potentially cure the disease. It would be hugely useful if you could have a an allo transplant, like off the shelf type of transplant, with a gentle myoablation, if you could move towards that, with cells that are resistant to HIV, I mean, it will be hugely helpful.
So Brigitte, just to clarify, you're talking about the patients who are resistant to HIV, these people, this population that are termed like elite controllers or something like this, or they're entirely resistant? Are those the same things, just for?
Well, there are some people that have a mutation in the CCR5 gene, a 32 amino acid deletion, and if you have that homozygosly, then you are resistant towards CCR5 tropic HIV. You're
Okay.
still susceptible to CXCR4 tropic HIV, which is less common. But the way that people have been cured is that they were identifying patients as donors that had a homozygous CCR5 deletion, and they took stem cells from them and used that as a transplant. But those donors are hard to find, and not only the donor has to be Delta 32 positive, which only less than 1% of the population has, but you also have to have the HLA match as well. I mean, which is even more important because, you don't want to get a rejection.
So could one of the searches now for COVID and thinking about repair or at least prevention in other cases be the search for these types of individuals who haven't gotten COVID or seemingly have had very limited or mild response? Is that a reasonable thought on cell therapy there? Versus, I mean, and would those immune cells, is it the immune cells that actually would need to be, would be useful? This is pretty speculative on my part since I'm not an immunologist.
I think for COVID, it will be mostly neurons, but it's hard to say what those people need because the symptoms are so non-specific, and if you give them new neurons, whether that helps or not, and if the infectious agent is still in you, the new neurons would then be also infected. I don't understand enough about long COVID, but I also don't think that the research is quite there yet, to say it's this or that. I mean, I think you would just have to try it out and see where that helps.
Reed, you had some thoughts here.
Yeah, just really quick. I mean, I think it's important. It's probably useful. I think it's a really great question. Like most of these viruses are like putting HIV to the side for a second. A lot of these viruses are acute, so you get them for 2 weeks and then they're gone. And now what we're learning is that's not the case, right? It lasts longer. You have these long-term complications, but you can almost reframe the question and ask, virus-induced tissue damage can, forget about COVID just for a minute. Are there synthetic biology approaches that can repair tissues? And then, which is way out of my expertise, but I think it might be more into others here. And so can you reframe it? Could it be that there's ways to use synthetic biology to introduce engineered cells or biomolecules that are going to repair tissue, and sort of maybe are those neurons infected to the side? Because if you can repair neurons or repair cardiac tissue, doesn't, independent of the infectious disease, and focus on that tissue repair, I think that's really exciting. Yeah.
The thing that we've been thinking about a lot with this is not just, so you either need to add cells, or you would potentially like reprogram and generate new cells from resident cells. And I think that people feel very uncomfortable about doing in vivo reprogramming because it's a little bit uncontrolled, and it seems very reasonable. But then the challenge becomes if you're thinking about doing delivery. So already for people with acute spinal cord damage, there are injections of iPSC-derived motor neurons into the the spinal progenitor, spinal motor neuron progenitors into the spinal cord. But the challenge of this is, of course, you're going to be putting them through a syringe into the spinal cord, and then neurons are not known for their affinity for shear forces. So the challenge there is even the delivery of how do you get something in and get it to survive. And so some of the more, I think, interesting things are, you can inject them or place them with some sort of material and maybe it protects them from shear. But there's limitations then on, even what you can inject and how fine a needle, these types of questions. And then also, can they get outside the material to actually then be useful? Other really interesting ideas I think people have come up with is, well, could you actually engineer cells to traffic to sites of damage? And that's very interesting because also sometimes damage is so distributed, you couldn't deliver into one single space and get an effect. And those effects aren't just like a surface, it's totally into different vascularized spaces, very thick tissues. And so I think that the idea of engineering cells to seek and find out sites of damage is also really interesting. We are very interested in doing this and developing what we're calling these search and rescue ideas, where they find damage and then you are actually mediating repair through those cells as well. But it's very sci-fi. So, oh, Chai-Yang, yeah.
Yeah, I promise I'll say something, but so I'm just listening to this like a topic 3 and 4 area. I just want to make a general comment, especially following what you just made a comment about engineering something, but sometimes there is always a concern on doing things in vivo because you just don't know where they are. I think this is where imaging, whether it is a molecular imaging or any type, I think the molecular imaging, which allows you to basically detect, visualize what's happening in the response in the cellular biochemical reactions inside the cells in vivo, intact or organism, is very helpful. And that is true for drug development, not necessarily the chemistry part, because that is a completely different thing in terms of how good the drug is. But it's the same thing, that when you deliver something into the body, mostly people do not know where they go, and they basically just look for the outcome, target the outcome. If there is some effect, good enough. But you don't, often you don't know what's happening systemically. Maybe there are some other things that are happening that you're not going to be able to do. So again, this is where imaging can come in. Now, the reason I said our cancer imaging program is getting very interested in incorporating or bring in SynBio kind of idea for imaging process is because since we're in the NCI, so we're very much into this cell-based therapy. But in itself, even with the success of CAR T cells for the blood cancers, still, for solid tumors, it's not doing very well. But at the same time, we always question, when you inject this CAR T cell, do you know where you have gone to? How much is at the site, and what is it doing? So there's a lot of research now going on, bringing in some of this circuitry design to try to make better CAR T, for example, smarter CAR T that can, so basically increase specificity and sensitivity of the cells. I think that applies to whether it's a cell-based therapy or drug-based therapy. You can, depends on the context of what question you want to ask. You can bring in imaging either as a way to detect a pathway as a response read of your drug in a target a more controlled manner, or you can try to design it so that the imaging component is a part of the drug design or delivery design. So allows you to know where the drugs went to. And then there are also examples where even the drugs go to different places, but including areas, locations where you actually want the drugs to work, you can actually use a smart imaging activated imaging as an activator so that you be able to activate the drug effect only in some places rather than for the entire body. So there are many different ways that you can play around. That's why I think a lot of times I listen to various cancer biology based research and drug development and screening research. I very seldom hear imaging as a part of it, other than maybe using imaging to look whether the tumor, like for us, like cancer, tumor is big or it's not there, or that kind of stuff. But that is very traditional. We need to move forward more to take more advantage of the imaging components whenever possible. But it is a context dependent though. Depends on what question you want to ask.
Absolutely.
My 2 cents.
Excellent. Excellent. So we're headed, we have like 7 to 8 minutes left. And I just kind of thought that the thing to close us out would be maybe the pandemic wish list of what we would want from synthetic biology. So we could get that, we've had a nice discussion. We covered lots of different topics and got to see sort of the preparation, the acute response, the long-term or maybe medium term of like, how do we look for drugs in a more effective way? And then thinking through how do we repair and will drugs be enough or do we need to turn to cell-based therapies? So maybe we could come up with a wish list of things that's like, if we were going to solve something, we can what would we solve? And we can go in order or we can just kind of go through, but I propose that we maybe go through orderly. and going through topic 1, 2, 3, 4. But I know that my brain doesn't always work like that, especially as we head towards 4:30 and it's dark outside. So
I think on the preparation side, sensing and identification, rapid identification, that's kind of critical. That was one of the big things I heard.
Yeah, that's great. I think Brigitte really nailed something with like, if we could have production chassis where, you're just popping in a new sequence and it's really well defined in terms of like what you're going to produce. And really it's just that surveillance. Like we have we have a very well characterized, vaccine chassis, whether that's protein based or RNA based. But we can put a new sequence in there and very quickly move towards all the screening that we would need to do to get that through, or actually, or whatever, or even thinking about what's the minimal screening that the FDA is going to require for some of these responses.
Okay, I think I have to sign off now. But I appreciate everybody's input and I really enjoyed the last 2 hours and thanks for all your time. And I learned so much with you, Brian, Teisuke, and Jian. Thank you very much for joining. I really appreciate it.
Thank you, Brigitte. Thank you so much.
Thanks.
So wish list, Christmas is coming.
I think you might be able to apply that to a couple of things here. I mean, I think you sort of touched on topic 2 there when you said when you want that minimal sequence, like we have a backbone for an mRNA vaccine and that's FDA approved, and if we could then print the epitope, quickly and not have to go through the process again, that'd be good. But I think speed and cost sort of is also an important thing there. I think I was brought up by maybe Brian early on for session 2 is can synthetic technology costs continue to go down?
Yeah, and maybe one of the other things we should think and we can think through is like, do we have, are there infrastructures that like things would become cheaper and faster if they were scaled bigger and we can build those now? Sometimes that's the nature of things. It's expensive because no one was doing it, but now everyone's doing it, so it's cheaper. So.
Yeah, I think for like LMICs, can we employ that technology in around the world? So it's not just in developed countries.
Yeah, absolutely.
Yeah, so scaling up here is going to help facilitate that.
I think on the response side, what Reed said was about the DNA synthesis, because that's a common thing that I've heard. This is the first time I've heard the synthesis part, but what I've heard from the other breakouts is, can we produce a lot of it? Can we insert it, and can it integrate into the genome? So this is kind of a third aspect to what we can do, what kind of our gaps are with DNA technologies.
So for us, in terms of like, if I wish list for circuits and cell-based therapies is, we want to make circuits really fast. We logically know how they have to perform, but doing the optimization is really hard, and engineering cells is like it's very time consuming. We have some ideas on how to accelerate the workflow, but part of it is actually just everything is so manually set up. Most cell line engineering is a function of grad student number. So that is inherently a fragile system. There's technology out there that exists that can make that a little bit more scalable. But it's still, I think for most academic labs, preclusively expensive. So there are things that so thinking through those workflows, like we could probably generate cell lines that could be useful for disease modeling, drug screening, but we can't do them at scale and then analyze that really fast. There's technologies again that are there, but and there's also I think there's actually there's a design aspect of how we're designing the circuits to allow for flexibility and plasticity that can be innovated on. But then doing that, we still have to validate, and that's, I think, with some of the stuff that's happening with microfluidics, and there's a marrying of opportunities there to make cell line engineering more rapid. If we could solve that, we could do really cool things, very, very exciting things. So yeah, that's my wish list.
Again, from one of the last breakouts, I think Wilson Wong said, cell lines, mammalian cell lines, if we had those developed and we knew characteristics, and unfortunately, no one's funding those right now because that's not science, right? But
yeah.
He said that was really critical for all of us, for the analysis and for the drug screening.
And part of that is also, I mean, this connects to the imaging aspect is, most of the time we characterize our cell lines, we're using fluorescent reporters. So it's an imaging technology normally that's used for the development so that we can actually QC things really fast in single cells. So I think it fits into the NIBIB mission within that aspect. But again, no one's like, yeah, we want to give you like a $4 million instrument to go make your cell lines. And I'm like, but I'll return good cell lines.
Yeah. We should make that the #1 priority.
Yeah, exactly. I was like, why is that everyone's favorite? I mean, actually it is. There's probably large equipment, pieces of equipment that, if you, that I think there's centers and stuff. I think I was talking with Jermont about this previously because it's my favorite thing to talk about. There's probably some large, like the Broad or something across the street might have these, but getting access to some of these cell line engineering instruments isn't trivial, and they're not. They can be high time use because the development of the cell line takes multiple weeks. So it's you really need quite a number of them to be able to pipeline things.
That would feed into antibodies as well. Sorry.
Yeah, exactly. No, 100%. So scaling our cell manufacturing abilities and taking the human component out of that is big. We can do this for yeast. I feel like that's one thing that makes evolution and yeast really work well, but we don't have that yet for mammalian cells.
That's very interesting. Imaging is something not my expertise area, but I studied some imaging technology development with the collaboration. And then that's another area my wife started to be interested in imaging because we use the diamond, whatever small diamond in the imaging in the component. But that's exciting. But one thing I also want to mention, actually I believe, or I don't remember whether I mentioned that or not. I mean, that's a little bit related, but a little bit different topic. But my wish list, we didn't discuss. We probably going to change our practice of currently cloning and engineering cell line. The point I'm going to make is we currently use antibiotic resistant gene for all the cloning, but the problem is that potentially contribute to the antibiotic resistant gene spread through horizontal gene transfer, but nobody thinking about that for almost centuries, because we just kill the cell and then dumping into the sink. But the problem is we blame hospital and agricultural people who use a lot of antibiotics, but we as a researcher also contribute to the antibiotic resistance spread because we dumping a lot of DNA that contain antibiotic resistance gene. So I want to get rid of those kind of practice. Actually, we develop something, but I'm not sure community kind of accepting those new practice, because using antibiotics and the screening and selection is so convenient. But I actually doing something, and I actually put some opinion article recently, proposing, getting rid of those kind of practice, but I'm not sure that will be accepted by the research community. But that's one thing I want to contribute to the society.
All right, that's the wish list. We got Tay-Sox. Well, we
Yeah.
need a couple more, or if you guys, Brian or Cheyenne, if you guys want to put anything in for the wish list, we're probably going to end our discussion in a few minutes. We want to make sure you have an opportunity.
Well, I don't really have a particular wish list, but other than just I would like folks maybe from time to time remember there's imaging tools that they should consider if they wanted to manipulate things in vivo, eventually move things to clinical practice, because you have to be able to know what you're doing. That in itself is actually very fundamental. There was actually a workshop, I don't know, a couple years ago, a bunch of therapeutic cell-based therapy, not just for cancer purpose. It's also regeneration medicine people. I forget the particular name of that workshop. They have a little article that written basically is like a wish list, and then things that feel what is missing. It is you're not be able to know what you're doing things blindly. That is a big challenge when it comes to translation, when it comes to going into clinical practice.
Excellent.
To be used. If you're just fundamental biology, that's a different thing. But as soon as you're going to try to say, I'm going to have some translational potential, then that means you're moving towards, treating something. Then it is a problem. So my wish list is just to hope that people don't forget that you need to be able to, sometimes seeing it is important to know what you're doing. It's not the only thing you need to do, but it will help. I think it will really help clean up and fine tune a lot of things that people are trying to develop preclinically or in the laboratory. And then even for the things that you want to design, like when you talk about circuitry, I want to do this ABCD in sequence, in parallel, those kind of things. That's all sounds very cool. But then if you start talking about, yeah, so one day maybe I can cure this disease, or maybe I can rebuild something else, then you are really talking about going into a living organism. And you really want to minimize so many times you do this blindly. That's
Yeah, that's great. That's so. Thank you so much, Cheyenne. All right, Brian. No pressure, man.
Okay. So I don't have a particular wish list, but I have some kind of future directions. I think might be interesting to kind of taking, as we previous breakout room already discussed, like evolution to consideration. Like think about ways that you can potentially drive the evolution of viruses. Like things like recently I'm thinking about like HIV. Like, is there any ways you can kind of drive the in-host evolution into less pathogenic And you can apply this perhaps like to influenza and other like highly mutable viruses. And recently I found like Emma Slack's work, who's from ETH Zurich. He's doing works on like kind of designing vaccines that can drive the evolution of salmonella. So I find that's very interesting, and there's a really lot of rooms to be done here. Yeah.
Brilliant. Awesome. Well, so this basically it closes our time and thinking about how synthetic biology can impact the pandemic. Thank you, Brian, again, also for, I like that. I'm so glad we had a grad student here to contribute too.
Yeah.
We want, I mean, there's so many diverse, everyone's perspective is a little bit different. In fact, I mean, that's the utility and the wonderful thing I think about the collaboration between, academia and industry and the government agencies, et cetera. So that's really exactly what we wanted to bring out, I think, from this this time. And now I guess my job is to get the summary of this prepared for our meeting next week. And then German, I'm going to turn it over to you just to kind of like, maybe close and reflect.
Yep. So I think if, Heather is going to push the last breakout, which is tomorrow from 12 to 2:30 Eastern, and you can see what they're going to talk about. And Heather, if you go to the next slide. And we're going to have our big powwow on the 14th, where we're going to hear from the breakout leads on their current work and then the summaries of the breakouts of today. And what I'd like to do is to thank Katie and Brigitte and Reed for leading this, because it was actually really interesting. The applied world, as I've learned, is always a little bit more interesting than the basic science. Because you can see where you're going, and we all know that this is a critical thing that we have to look at for the future. And with that, we can end the session, and I thank everyone for their participation, and thanks to Infinity for helping with the hosting. And again, I hope you have a good night, day, wherever you're at, and we'll see you at the main meeting.
Bye. Thank you.
All right, thank you very much.
Thank you.